Hello. Cell division is actually about how the cells divide themselves accordingly to either form normal body cells or to form cells that are specialized, and the specialized cells are what we call the gametes, which are involved in sexual reproduction. We get some facts clear again first before we go on. Pay attention that this is just a summary, and the salient points are highlighted here with your common errors and things that you don't understand. You still have to read through the notes to get the ideas clear. So one of the common terms that we always need to define would be the term homologous chromosome. So like I said, this basically this term of homologous chromosomes. Homo basically means the same, and homologous chromosome, the logos means same length, alright, same centromere position, and if we can add on would be the same gene loci, which is the same gene location carrying a certain trait. For your information, once again, for this and this could be two different genes, one with a big A, one with a small a. These two are what we call the alleles, being the alternative forms of a gene. Homologous chromosome basically, like I emphasize again, it would be the same length, the centromere position, alright, and as well as the same gene location, same gene loci will be a bit easier word to remember. Okay, I mentioned that for a reason, because we are coming to the human chromosome map soon. Basically, haploid here, the word say haploid, HA means half. Diploid basically means the normal chromosome number of a human being. And basically, an organism always produces gametes. The aim of it at the end is to maintain the same number of chromosomes, therefore actually maintaining genetic stability. Gametes are basically the sex cells, meaning the sperm nuclei, as well as the ovum. Because 23 plus 23 will give us 46 chromosomes, which is a normal number of chromosomes in the human cell. Haploid basically means a single N, and diploid means double N. This idea of the N and double N can be further explored in other creatures such as plants. A typical example of this is basically 6 here, as you can count. First, to remind you once again, if you count the centromere, what are we actually counting? Are we counting the amount of the DNA, or are we counting the number of chromosomes? Counting the centromere is counting the number of chromosomes. If we count the number of sister chromatids or number of chromatids, then we are counting the amount or quantity of the DNA. Looking at this little nucleus here, you can see that there are 6 of them here. 1, 2, 3, 4, 5, 6. And basically this example here shows you the sister chromatids on a duplicated chromosome, because both of them are the same. So we call them sister chromatids. Non-sister chromatids occur between two different pairs, like the blue and the red color one. These are non-sister chromatids. They are not sisters. And the centromere position is usually at the center here. Then by drawing here, it will be easier to see. And 2 of which makes up a pair of homologous chromosomes like this. Okay? Back to the human karyotype map. This is the human chromosome map, which you will see from time to time. Once again, the most important thing that we are able to see here, the information that I actually get from here, would generally be number 1, the number of chromosomes we can see here is 46. We can also be able to tell that each of them are just homologous chromosomes. And the reason why they are in pair, because one of them is from the father, the other one is from the mother, and it's important for you to know that. Plus, we are also able to see the gender of the person based on the human chromosome map. XX meaning that this will be a female, and if it's an XY, it will mean that this is a male. So be very extra sensitive when you're looking at the chromokaryotype map, because it may also tell you mutations to get take place. That's the famous one in 21. This person does not have Crown syndrome because there is no extra chromosome at chromosome number 21. Moving on, we're looking at the interphase. Basically mean, as we have said before, is actually for cell growth, as the longest period of a cell growth is dedicated to cell growth, and it accumulates raw materials for DNA synthesis. If we officially define DNA synthesis, it's basically the DNA in the chromatin which condenses to form sister chromatids. And the chromatids are identical after DNA replication has taken place. So generally speaking, this is one chromosome, and before anything happens to them, they look like that. So after we've gone through the DNA replication, you will realize that there is actually one more sister chromatid, and this we call DNA actually times 2. Okay, so generally now the idea now you should be very clear how do we actually explain what is DNA. Is basically found in chromatin, which will condense to give you sister chromatids during interphase. And the chromatids are basically sister chromatids because one of it has become 2 during DNA replication. And officially, how we count the number of chromosomes, so we look at the number of the centromeres that we have. Moving into mitosis exemplar, the importance of mitosis and the purpose of mitosis is basically to actually make human cells. They have the normal number of chromosomes 46. You have to read the rest of it in the notes as for the purpose and the importance. The idea about prophase here, we look at the diagrams of both of them, is the very easy to remember will be the 2 C's. Sorry, one of it is wrong. The 2 C's here that we are looking at, and the other one that we are looking at is basically the 2S and the 2N. 2 C's will be the chromatin condensed in the chromosome, the centromeres moving to other parts of the nucleus. Not nucleus actually, this one is basically it starts to move to the opposite side of the cell, and the spindle fibers actually form. Then therefore the spindle fibers will attach, okay, the centromere, okay, together with the sister chromatids. So they look like that. Alright, and first we need to know that, for example, we need to treat the DNA and the nuclear material. Your nucleus must disappear, and after that followed by the nuclear envelope breaking down. Take note that for mitosis, this stage we are dealing with splitting the cell equally into half. To split the cell cytoplasm equally into half, it's cytokinesis. So if a diagram that looks like this, you can look carefully. There's a lot of disintegration, a lot of condensing, and the nucleus here, if it's pretty much intact, it is actually prophase, but maybe a little bit earlier. If it's not very much and still look like this with a lot of holes at the end here, then probably it is actually breaking down. If you're looking at this easier to recognize diagram with actually holes here, and you can see that there are 2 of them like this. It means DNA replication has taken place. Alright. To metaphase simply basically means that the centromeres of the duplicated chromosome, they are already duplicated, they lie across the equator, and you can see they're going across the equator. You will see that there is about a certain plate that is happening here. Once you see such a diagram and they are arranged in pretty such a nice way, we say that they are actually aligned nicely across the equator of the cell, and each of them is connected to a spindle fiber again at the centromere. So basically this is lining up first. This lining up is important because it prepares them to split later up. Anaphase will actually look at things about the separation of the sister chromatids. As you can see clearly, every one of them, after they are separated, you will see clearly that the centromeres will divide. Please do not never say that it splits into 2. That increase the centromeres have actually and been split into unequal portions. Dividing means that the centromere have actually been divided and equally into 2 parts. And we see that the chromosomes are separated into two groups near each pole. Looking at these, you'll be able to tell there's a lot of spindle fiber movement, and they are actually aligning at two parts of the opposite sets of cell. Take note that if you're asked to draw the V shape, the moment when you pull in this direction, the V shape, the V will always be, the broader end of the V will always be facing outwards. Please do not draw the ends like this. Please do not draw the ends like this. Pretty scary if you draw it like this. And this spindle fiber is pulling here, then this is exactly wrong direction. It will never be like that. Alright? Telophase will be about the end of the mitosis stage. As we can see, everything here will reverse. It will be the reverse of your prophase. Chromosomes will start to uncoil and they become chromatin backwards. Why do they need to go back to chromatin again? Because they are no longer dividing anymore for now, because your nucleus will become visible again as the nuclear envelopes will reform. Spindle fibers will break down. For those that are not really required, they will break down. And cytokinesis, please take note that it takes place simultaneously. It is not one stage after the other. As you can see here, it is an animal cell. You see that there are things that are happening here, and for a plant cell, there is also something happening here. But stereocytokinesis is looking at the formation of a new cell wall in the center of the two cells that will split the cytoplasm. Please remember, alright, equally between the two daughter cells. As for this one, it will just look like a bar here. Basically, this is the formation of a cleavage furrow that will split the daughter cells for the animal cells equally into two. Again, the cytoplasm. Meiosis is a reduction division, and the reduction division simply means that the chromosome number, please take note, is reduced by half. Through the separation of the homologous chromosomes, meaning to say for human beings, you will go from 46 to 23. And there are two consecutive divisions. Consecutive simply means that if meiosis 1 takes place, and following that, after that, meiosis 2 will take place. And the total number of daughter cells that will receive are 4, and they are all genetically unidentical. Prophase 1 or meiosis 1 is a very special stage. During this stage, chromosomes coil and they will condense, and there's a pairing of homologous chromosomes that will occur. Each chromosome has two chromatids, as we mentioned before. To actually remove the genetic material and cause the pairing up, the nuclear envelope must break down first. Centrioles will move towards the opposite poles for animal cells only, and the formation of spindle fibers. Let's take a look at a special event that is here. Event special to only prophase 1 is the pairing up first of the homologous chromosomes, like this. This process is termed as synapsis, following which synapses have actually a pair up, and this crossing over will take place. The points of information that happen is actually at the chiasmata. Chiasma is singular and chiasmata is plural. So crossing over chromosome segment always exchange between a pair of homologous chromosomes. This is a pair of homologous chromosomes, and this is another pair. Alright? And as a result after which they have actually exchanged it completely, they will result as recombinant chromatids. But we don't really use this term nowadays, so just take note that just remember that prophase 1, this will actually result, because for this stage it is further down. Meiosis 1. We're just taking a look once again what happens here. You can see that there is synapses happening, and following which synapses happening, the crossing over is taking place. Following which the crossing over has taken place, the genetic material and also the chromosome segments are exchanged. So they are no longer genetically identical. Alright, So just to emphasize the idea, alright? Once everything's ready in prophase 1, the lining up of the homologous chromosome pairs. Alright, and at the equator of the cell, as you can see, the equator of the cell has changed. Meiosis and mitosis. The equator was here. So for meiosis, the equator has rotated 90 degrees. And you can see here clearly for this one. They are still together. They have not separated yet due to crossing over. Alright, so please take note that spindle fibers were then attached to them, each of them here 1, 2 to the centromere of each of the chromosome. Following which, during metaphase 1, we also like to emphasize one very important event that is happening that gives you variation. The first event that gives us variation was crossing over during prophase 1. So this one, we call it a little bit different. It's actually much longer than that. This we call it independent assortment of homologous chromosomes. Meaning to say, they will line up independent of each other, okay, randomly at the metaphase 1, the equator of the cell. Each pair of chromosomes, maternal and paternal homologous chromosomes, okay, they will be assorted or they will be separated into different daughter cells independently of the other pairs, like this. Okay, as you can see, the possible arrangements have been actually arranged and shown here, the differences being highlighted here. Alright, so just remember that this is what we meant by the independent assortment of the homologous chromosomes. Once again, to show you the possibility arrangements of the same thing, it's one possible arrangement here, what's happening, and you will get to see that the arrangement finally at the daughter cells are extremely different from each other. You may be asked to draw the combinations of the chromosomes that will occur, but do take note that whatever you draw, you need to make sure that you start from the metaphase 1 arrangement given here, okay, and finally metaphase 2, which will result in the different combinations of the chromosome at the daughter cells. At anaphase 1, when things start to grow apart, please take note that the centromere is still here. The centromere is still here. And there is no dividing, and that the spindle fiber will simply pull it apart to two different cells. Also take note that at this time when your homologous chromosomes separate, you will get 2 chromatids per chromosome. This is unlike your mitosis. Telophase 1 takes place when half the cell has a haploid set of chromosomes, meaning to say N. Alright? And each chromosome still consists of 2 sister chromatids, like this. Cytokinesis will always take place simultaneously. Looking at this cell is obviously an animal cell with the formation of a cleavage furrow once again. When we go on to prophase 2, when cytokinesis has almost completed, please take note that there is no interface between meiosis 1 and meiosis 2, because meiosis 1 we have reduced the number of chromosomes, but we have not reduced the DNA quantity. So to reduce the DNA quantity, we cannot carry out interphase again. If not, we will double the DNA quantity again. Remember that? And so the same thing goes again, spindle fiber forms, and chromosomes will still have 2 chromatids, same again, move towards the metaphase plate or the equator. If you look carefully at this meiosis 2, it is very similar to mitosis. Metaphase 2, we see this happening again. Almost everything is the same as that of a mitosis, but you realize that they are just genetically identical. Sister chromatids are genetically identical. So this is one of the differences you can quote as well. Alright. And next one, and the phase 2, we realize to see that there is 2 newly formed chromosomes that are pulled within the pole. So as you see it, now the centromere will split. Okay. Now it's going to be pulled this way. And after we have done it for telophase 2, the completion of the process will be when the chromosomes have reached the poles, everything will be opposite to phase 2. Nuclei will form, the nuclear envelope reforms, the chromosome begin to decondense as well. Cytokinesis will begin to split will begin to actually split the cytoplasm into 2. Take note that this one was actually from meiosis 1. Alright, and if sometimes in pictures you will see like this, it means that meiosis 1 has completed. It doesn't mean that you don't get 2 daughter cells. Now the cleavage furrow will form this way. So in doing so, you actually split the whole cell into 4 daughter cells. So diagrams may differ a little bit. Alright, as you look at the DNA amount now, just now instead of 4, now we have only 2. Okay? So to summarize our whole process once again, the process of it is from the pairs of homologous chromosomes in the diploid parent cell. During their duplication, when they go through the duplication and they go to meiosis 1, they will separate. So meiosis 1 is reduced chromosome number. Meiosis 2 is to reduce the DNA quantity. Take note that the differences between meiosis 1 and 2, before meiosis 1 there's interphase, before meiosis 2 there's no interphase because the DNA amount needs to be reduced further, so you cannot double the DNA amount again. So we're going to emphasize one last time before I close the video, the origin of the genetic variation that we have here. Okay, so we are looking at the behavior and it's different. The behavior of chromosomes during meiosis and fertilization is different. So this gives rise to our variation. And variation basically means the independent assortment of homologous chromosomes, is what I mentioned just now. The crossing over during prophase 1. Independent assemblage was metaphase 1. Random fertilization was the random combination of the gametes that take place during fertilization. Please take note that during gamete formation, meiosis takes place. To form many more sperms, you need mitosis of the sperm that is actually formed. For the ovum, there is no mitosis at all. You only produce one ovum from one ovary during ovulation, during the menstrual cycle. Okay? And so that's it. If you still have any more queries, please, you have to ask. Just clarify for you. Thank you.